I'd just like to start with this conflict of interest disclosure. My husband is Albert McGuire, our co-authors on a patent that was recently awarded for a method of treating or retarding the development of blindness, but we waived any potential financial gain more than a decade ago. So I'd just like to start out telling you these studies have evolved from a really fantastic clinical trial that we've been carrying out thanks to the invitation of Dr. Catherine High, and the results for his single injection to his worst eye is seen navigating with a cane and holding his parents' hands, whereas after injection he can sit in front of a classroom, he can play baseball, he can ride his bike around his neighborhood by himself, and see things in dim light, which obviously he couldn't do beforehand. And so the results have really been extraordinary, particularly with children with this disease. And we've continued with this clinical trial, as I'll tell you a bit, but it's also evolved to include some very interesting scientific questions. And so what I'm going to focus on today is a study that we've done in conjunction with Dr. Manzar Ashgari, who's shown down here at the bottom, and her research associate, Laura Sykowski. These are two separate teams here. The gene therapy team at Children's Hospital of Philadelphia runs the clinical trial and plans ahead for what's going on forward and takes care of the patients and so forth. And that is a team led by Dr. Catherine High, and the PI of the study is Dr. Albert McGuire, somewhere in the back here, yes. And we have just made, you know, it's a kind of village of people running this with great clinical coordinator and regulatory people and vector people, et cetera. The CNS imaging team, again, just does the studies of the brain imaging, asking some very interesting questions. So, for example, it's really not been known what happens to the brain when somebody's blind. So vision depends, of course, upon the retina being able to see things, but that has to relay the signal to the brain. And in many cases, it's been thought that the brain is the part of the brain that understands vision is taken over by other cells, for example, auditory cells. And so it's not known what the extent of recovery is, the recovery potential is of the visual cortex after people have been deprived of vision for many, many years. And so the studies here aim to look at these questions: age limits for rescue, how long does it take to get rescue, how stable is the effect, etc. And so just as a little background, this disease is congenital blindness. It's a rare disease. There are approximately 3 or 4 babies born per year in the United States with this disease. It's usually identified first in parents because these kids aren't fixing and following and responding the way normal sighted kids do. And whatever vision they have disappears with time, shown here in visual fields. They do have light perception at birth, but but by the time they're in their late 20s, 30s, they're very severely impaired, and their vision in childhood is abnormal, as I showed you in that first video. So what is the approach we're using? Well, first of all, the gene defect that we're studying is involved in recharging vitamin A that's necessary for the photoreceptors to be able to absorb light and send the signals to the rest of the cells. And it's, this process is helped by an isomeral hydrolase called RP65, which is present in the retinal pigment epithelium. This helps deliver the vitamin A molecule called 11-cis to the photoreceptors. And when this is mutated in this form of the disease, that vitamin A molecule can't be delivered, and so there's blindness. So the idea is really simple: to deliver the normal copy of the RP65 cDNA to those cells and to correct the biochemical deficit. And that is done as shown here in the clinical trial with a construct which delivers the RP65 cDNA packaged in a recombinant adeno-associated virus, delivered subretinally to the area in the retina which the surgeon deems is most promising in terms of restoring function. And these studies have built upon many years of animal studies, including help from this guy here, shown here, who was the first dog to receive and benefit from gene therapy for this disease. So this is a summary of our phase 1/2 study, which began in October of 2007. Enrollment was completed in June of 2009 of 12 individuals. These individuals are all legally blind, 8 years old and older. They had to have RB65 mutations, otherwise the treatment wouldn't have worked. And it was a dose escalation study shown using these doses. And the safety profile was excellent. There's been no severe adverse event related to the vector delivery at all. So it's been very exciting, and the subject's been followed now for more than up to 4.5 years. So now we get into the question about looking at the brain, and the way that this was evaluated by Manzar Ashtari and Laura Zajkowski was to take advantage of what's called BOLD, blood oxygenation level dependent functional magnetic resonance imaging. And simply, this is MRI imaging, but it takes advantage of the fact that you can see where there's high level of blood flow because there's iron in the red blood cells, and that appears very strongly in the magnet. So this shows the setup with one of our subjects with a magnetic coil around their head, and they're wearing goggles. And in those goggles, they are presented stimulus, visual stimuli in one eye and the other. And these are what the stimuli look like. They're flickering checkerboards, some of high contrast, some of medium contrast, and some of low contrast. And they're presented somewhat randomly from one eye to the other with rest periods in between. And what this study does is take advantage of the very well-known connections. There's essentially a wiring diagram, explicit wiring diagram between the retina and the visual cortex, which is in the back of the brain. So just to give you an example of the results and illustrating the power of this wiring diagram, these are two subjects. This was the boy I showed you earlier, who was injected in his left eye. The blue areas show his visual field before he received his injection in both eyes. You can see they're very small. Normal-sighted people have visual fields that extend over this whole area. And after injection, which was in his central retina, his visual field expanded in this amount shown in red. And so then when he was, his right uninjected eye was presented with this flickering stimulus, he saw barely anything as judged by the fMRI. There's hardly any signal here. Whereas when his injected eye was presented with this signal, you can see a robust activation, which maps almost explicitly to this area that was injected. And just shown for contrast, this is an older person who received a more peripheral injection, and you can see in his injected eye, you can see the visual field is expanded in this area, more lateral. His central retina wasn't injected because it had degenerated due to the disease. You can see his activity is in the lateral part of the visual cortex, mapping to the retina, just where it would have been predicted. So, this data was really exciting and showed that it was possible to activate these pathways from the retina to the brain, even despite decades of significantly impaired vision in these patients. So the next question was, is it possible to treat the fellow eye safely? And this was being done by the clinical team. And if so, what happens as far as the brain being able to understand this? So this is what the clinical team did. It did some re-administration studies. We carried out studies in large animals, including affected dogs and monkeys, and many other studies. And we went and proposed to do a study, a re-administration study in our subjects, and were allowed to first carry out the study in 3 adults with an 8-week stagger, so that they could be monitored for immune responses. Because the main concern was that this first injection would be like a vaccination, you'd come back with a second injection, it'd be like a booster shot, And what we don't want to see is inflammation that would wipe out the benefit that they had received in the first eye, and we don't want to see an immune response which would prevent benefit in the second eye. So these 3 individuals participated, and each one of those gained vision, and there were no safety, significant safety problems identified. So after a review, we've gone on to inject the remaining eligible subjects in this clinical trial with a reduced stagger in between the subjects. And that's where we are now. We haven't finished that re-administration process. But we have injected 8 eyes of these 12 individuals. So let me just show you the view from the clinical team. This is an eye of one of the children who received a second eye injection 2 weeks after injection. And it's a clear view through the vitreous to the back of the eye. It's very hard to see where the borders of this injection were. In fact, that's a hint. That's where the retinotomy scar is, where the cannula went in to inject. And this is a view through the operating microscope from a video taken at the time of surgery with a little bleb of high-density liquid over the optic nerve and fovea. And you can see the borders of this bleb, which are here, where I put these arrowheads. There's no sign that there's been any damage done here. So then what happens with the brain imaging? Well, I just want to show you how the rest of the image analyses were carried out. So essentially what I showed you before was a flat, a section through the brain. But the way that this can be analyzed better is by taking advantage of the 3-dimensional array of the brain, and to eliminate the problem that the brain has valleys and peaks and so forth, to essentially blow up the brain with computer software, and then to snip it and tuck it and flatten it out so that you can see the areas. And let me just show you what the results would look like in a normal-sighted individual was presented with the stimuli that I showed you. When this blown-up brain or flattened brain is illuminated with light, you'd see activation in these regions which map to the visual cortex in the back of the brain. So that's normal. So here is the result from one of the individuals who was evaluated at baseline with fMRI in the second eye. You can see over here, there's no sign of activation in the visual cortex. By day 30, you can start to see activation, and then by day 90, you can see very strong activation in the predicted regions of the visual cortex. And that timeline is found in all the patients we looked at. Basically, it takes about 3 months for the brain to really kind of level out and plateau in terms of responding to these signals. Now, what about the initially injected eyes? So these are the initially injected eyes of all 3 patients. You can see there's some signal there because they've already had 1 injection of this material. And here's the surprise. 90 days after administration, re-administration to the second eye in these individuals, you see very strong activation after their initially injected eye is eliminated. This was an unexpected finding in that the previously injected eye continued to improve after the injection of eye number 2. So we thought about this for a while and looked at all sorts of additional data and realized that one explanation that is the diminution of nystagmus, the abnormal eye movements in these patients. So shown here is one of these individuals at the baseline before having a second injection, and then 90 days after having a second injection. And you can see that there's still some wobble in the eyes here, but it's really diminished compared to what it was at baseline, where it was really wandering all over the place. So anyway, that's a hypothesis, something that we're still looking at, but intriguing nonetheless. So just to sum up, we've shown that the neurons in the visual cortex are still responsive even after decades of severe impairment, that the gene therapy renders both the retina and the brain more sensitive to light, and I didn't tell you about contrast, but it does render it more sensitive to contrast. The retinal treatment area correlates roughly with the area of cortical activation, And there's good correlation between our psychophysical tests and the results from the fMRI. We think that it's possible that fMRI will play a potential role in the future in screening potential subjects who could potentially benefit, and this could also be an outcome measure for measuring some of the efficacy, and certainly be used to probe variables affecting vision and CNS plasticity. And these are results from one of the children in our study. You can see that this is really quite a dramatic response here. So finally, in addition to our team members, I'd like to thank the subjects in the study without whose help we would not be able to make this progress. Thank you very much.
You raise a really interesting point, which is that you don't have vision for many years, even though you stimulate the visual function. You believe, though, that the patients can get to interpret what they see, because the neurons are still there and can be educated many years later.
It's fascinating, actually, particularly in thinking about things that patients have told us anecdotally. So, for example, oh, I looked at her and I didn't know until 3 weeks after the surgery that she had blonde hair, you know, that kind of thing. Or, wow, there's a tree branch outside of my window. So, like seeing something and then putting together what that thing is. So I think we're also seeing some sort of synaptic plasticity. Hard to explain what it is, but I think this will be a handle towards understanding that kind of biology.
That's very exciting. So what are the retinal diseases you think would be next up with this approach?
Good question. I think other forms of Leber's congenital amaurosis are good targets. There are several which are heading to the clinic already. Choroideremia is one that had started in the clinic about half a year ago, and I think there was a talk here at this meeting about it. Stargardt disease and macular degeneration. There's some technical challenges with each one of these, but I think that they are addressable.
So I saw that on the imaging at 90 days that it sort of reaches, I guess, its peak. When do patients tell you, now I'm seeing, and that's kind of when they're having that sense of clinical effectiveness?
Well, interestingly, they notice a change really early. By about 8 to 10 days, they start noticing everything is brighter. But the brightness doesn't really coincide with the objects. So they describe things being brighter about 8 to 10 days, but by, by 1 month, they can actually see forms better. And by 2 to 3 months, can tell things like whether somebody is smiling or grimacing or frowning, details which are a lot more subtle than shades of white and black. So it's a process, but by the time they're 3 months out, that's a time when we've been able to correlate the sort of plateauing of some of our other measures. For example, improvement in pupillary light reflex or light sensitivity.
Jean, this is really great. So you've treated subjects at different ages. Do you have enough data to compare if there's any difference in terms of response rates according to age? I mean, do older folks respond less than younger folks?
That's a hard question to answer because with the age, there's degeneration, so that the older subjects have more degeneration, have less retina, basically, to respond. So we don't really have the controlled situation with a young person and an older person in the same level of degeneration. But maybe we'll see that. I don't know.
Also, you said about 3 or 4 babies are born with this a year. So now, and that the parents pick up that there's a problem pretty soon, right? Mm-hmm. So now, for next year, for those 3 or 4 babies that are born, are they going to find their way to you, or is there some kind of a plan?
We are planning a phase 3 clinical trial, again under the direction of Catherine High, at 2 centers, at Children's Hospital of Philadelphia and also University of Iowa, with ages 3 and up. So we're reducing the lower age limit. with the hope that we can essentially prevent the problems of the disease and allow people to have normal or as normal vision as they can from early in life. Ultimately, we think the best results would be if babies are injected at the time of diagnosis, but it's sort of a stepwise approach going to younger and younger ages because of risk-benefit issues and surgical approaches and so forth.